In this video, we're going to review the cell cycle and mitosis, but add a few more details to it. You've seen this in grade 10, and again, we're just going to add some more depth to it with a few more terms that you need to know in the grade 11 course. So the purpose of mitosis, again, is to help you grow, to help you repair yourself, and help maintain the organism. I just want to go over a few big ideas first. So for the cell cycle and mitosis, the parent cell, the original cell, is diploid, starts off with 2N. And the end result is that the daughter cells, the cells that are produced, are also 2N each. They have the same number of chromosomes as each other, same number of DNA as each other, same type of DNA, same codes and everything as each other, and also they're identical to the parent. So they're basically copies of the parent. It's like the parent photocopied itself. So the end result of the cell cycle and mitosis is you have two daughter cells that are identical to each other and to the parent that was used to produce them. So one of the big ideas of this lesson is understanding what happens when you grow, when you go from baby to toddler and then from toddler to you, or when you go from, well, not you, but when a plant goes from seed to a bigger plant to an even bigger plant. Is it that you're getting more cells or that your cells are becoming larger? Which one is happening? Or is there no change at all? So you can pause the video and think about that. What happens when you grow? Are your cells just getting larger? Are you getting more of them? So I want you to understand that you're actually getting more cells. Those cells are dividing by mitosis to make more of them so you can grow. Your cells aren't actually getting larger. It's actually a bad thing if cells get too big. To survive, cells need to be pretty small. The bigger you get, the more difficult it is for the cell to, well, let's just say feed itself. Nutrients come in from the outside and enter through the membrane. So if a cell is small, the nutrient comes in and can make its way through the entire cell, including to the middle, pretty easily because there's not much traveling to do. But if a cell is large, then it takes a long time for the food to enter and then go to the middle. Or if garbage is produced, it takes a long time for that garbage to leave the cell, and so the cell could die because it doesn't get enough nutrients or get rid of waste fast enough. So ideally, cells should stay small. That's why rather than your cells getting bigger, rather than you becoming a blob, a big giant blob, your cells make more of themselves and stay small, which is what allows you to grow. So your cells stay small, but they just make more of themselves so you can grow. Otherwise, these cells would die. The idea is that a cell wants to have a large surface area, but a small volume. Large surface area, so lots of membrane, but a small volume. It wants lots of membrane because if you have more membrane, more food can come in. But it wants a small volume so that the food doesn't have to travel very far. So for example, if the cell is flat, the food doesn't have to travel very far, so the food can enter all parts of the cell. This is again why your cells do not get bigger, but rather they get more numerous instead. So that's the idea behind the cell cycle and mitosis. It makes more cells, not larger cells. So the way you become you, this is a story. Your father donates a sperm, your mother donates an egg. The sperm cell has 23 DNA molecules in it, or 23 chromosomes, and it's haploid, so we write N is equal to 23. The egg has 23 chromosomes and it's haploid, so we write N is equal to 23. And that produces a single-celled organism called a zygote, which is now diploid, meaning it has two sets of chromosomes, a set that came from dad and a set that came from mom. So now it's 46 chromosomes, 23 plus 23. Now you don't stay one cell for your life, and that cell doesn't get bigger to make you. Instead, that cell will go through mitosis and make more and more and more of itself. And all of these cells here will continue to be diploid. They will continue to be diploid. When the sperm and the egg join each other, that's called fertilization. The sperm fertilizes the egg. Fertilization. And so what we're going to do is we're going to focus on this part here where the cell cycle and mitosis make more cells that continue to be diploid. So you, at one point, were a unicellular organism made up of one cell, unicellular, and then through mitosis, repeated rounds of it, you became multicellular, made up of many cells. So just a quick review before we move on to the cell cycle and mitosis. Remember that humans have 23 pairs of chromosomes. That means 46 chromosomes total. 23 chromosomes came from dad. They're called the paternal set. 23 chromosomes came from mom. They're called the maternal set. So that means you have 2 sets total, 46 chromosomes total per body cell. A body cell is called a somatic cell. So that's things like cells of the liver, the heart, your skin, and those cells are diploid, meaning 2N, diploid. A cell that's not a somatic cell is called a sex cell or a gamete, and that is haploid. It has 1 set of chromosomes, haploid. We represent that with the letter N instead of 2N. So this is diploid, and this is haploid. And again, why are we talking about chromosomes? Well, that's because chromosomes have your DNA. Chromosomes are basically just your DNA wrapped around proteins. So all your genes are found on these chromosome structures over here. And so when you're making copies of your cell, it's important that you copy that DNA exactly the way it is. Otherwise, there will be problems with the cell along the process. Now remember that sometimes your chromosome will be in the form of chromatin instead, and we're going to take a look at when that is in just a bit. So here we have a picture called a karyotype. A karyotype is just a picture of your chromosomes in a somatic cell. And the reason I know this is a somatic cell is because there's 46 chromosomes. If I had 23 chromosomes in the picture, that would be a gamete. So this over here is 1 chromosome, this is another chromosome, this is another chromosome, this is another chromosome. This is another chromosome here and here. So you'll notice that they're organized in pairs. There's 23 pairs total, which means 46 chromosomes. Now the cool thing about this picture is that in each pair, 1 is from mom and 1 is from dad. 1 is from mom and 1 is from dad. Mom, dad. So that's why we say you have 2 sets of chromosomes, 1 from mom, 1 from dad. Now just by looking at this picture, I know this is a boy, and that's because there's a special set of chromosomes over here called the sex chromosomes. If you have XY, it means you're a boy. If you had XX, that means you're a girl. We'll see more about that later on. This is just a lame joke. Hopefully you get it. So another important term we need to discuss is something called ploidy, and I've been discussing that on the previous slides. Ploidy just means the number of sets of chromosomes that you have. So if you have 1 set of chromosomes, you're represented by the letter N, and that's called haploid. So typically gametes, like the sperm and the egg, another word for the egg is the ovum, or ova plural. Ova plural. They're haploid. They have 1 set of chromosomes each, and in that 1 set for humans, they just happen to have 23 chromosomes. So it's 1 set of 23. Whereas human somatic cells are diploid. Diploid, meaning they have 2 sets of chromosomes. 1 set of 23 from mom, called maternal, 1 set of 23 from dad, called paternal, to give you a total of 46 chromosomes. To represent diploid, we write the letter 2N. So humans are 2N is equal to 46. But other organisms have different numbers of chromosomes, so you can have some diploid organisms that have 2N is equal to, let's say, 14. And so their gametes would be N is equal to 7. But it's the same general idea. Diploid represented by 2N, haploid represented by N. There are other types of ploidy out there called polyploidy, which means you have more than 2 sets. You can have 3 sets, be a triploid, 4 sets, and be a tetraploid. That's usually more common in plants than it is in animals, although there are some animals that are, for example, tetraploid. So we'll do one sample problem together just to make sure you understand the terminology. So we'll do this sample over here. If an organism has N is equal to 14, find out how many chromosomes the organism has in a somatic cell and in a gamete. So for this question, you look at it carefully. N is equal to 14, that's the haploid number. So if that's the haploid number, that's how much it has in a gamete. In a gamete, it would have 14 chromosomes or 14 DNA molecules. In a somatic cell, that's diploid, it would have 2n is equal to 28. In question B, it's similar, except I give you the diploid number to begin with. So an organism has 2n is equal to 14. Find how many chromosomes there are in the somatic cell, which is the diploid one. So that would mean this is 2n is equal to 14. But if you want to find the gamete, you'd have to find the haploid number, which is n is equal to 7. So I'm going to leave question C for you to try on your own. But what I want you to do is do an example of drawing a fertilization event. So I'm going to do the fertilization event for part A, meaning draw a sperm from the organism A, fertilizing the egg from organism A. So what I would do is I would just draw the sperm like this. I would write n is equal to 14. I would put a plus sign. I draw the ovum. I put n is equal to 14 because the egg also has 14 chromosomes. And then I just put a little arrow to show that it made a zygote. And I'd write 2n is equal to 28. And you can label that sperm, ovum, and zygote. And you could know that these are haploid, haploid, and diploid. So that's the general idea behind drawing a fertilization event and figuring out your diploid and haploid numbers. So now we're going to just take a look at some more important terminology before we go into the details of the cell cycle. The parent cell is the original cell, the cell that you start off with, and the new cells that are produced, there are 2 of them, they are called the daughter cells. And so essentially what happens is mitosis, you need to understand it preserves ploidy. That means if the parent cell started off as 2n, the daughter cells will also be 2n. So these are the 2 daughter cells here, they would be 2n each. And then these 2 daughter cells, they could eventually become parent cells, and then they can produce their own daughter cells. And they would also be 2n each. They'd have the same amount of DNA, chromosomes, and same genes as the original parent that made them. I also want you to get that mitosis happens in somatic cells, not in sex cells. It's in somatic cells. So skin cells, heart cells, liver cells, not in the gamete-producing cells, not really in the germline cells, which is gamete-producing cell. Mitosis is a form of asexual reproduction, meaning that it only requires one parent and it produces identical offspring. So when you get cut on your hand, if you want to heal yourself, you want the same type of skin cells that were there before. So cells in the surrounding area will make more cells to replace the cells that were damaged in that wound. And again, it's from one parent. So all these, let's say this was a human cell, all these cells, when they divide, they're going to produce daughter cells with 46 chromosomes each. So you'll notice there's a pattern here. After the first division, there's 2 cells. After the second division, there's 4 cells. If this division continued, you'd have 8 cells if there was a third division that happened. So our general formula is 2 to the power of n for the number of divisions. So for example, if there's 1 division, well then there's 2 cells that are produced. If there's 2 divisions, then the end result is 4 cells that are produced, which is what you see in the picture. But if each of these produces 2 cells each, that would be 8. So that would be our third division, 2 to the power of 3, which is 8 cells total, and you could continue that. One of the most important points, though, is that mitosis preserves ploidy. You're going to see another type of division later on called meiosis, and that one halves the ploidy. So if an organism was 2n, the meiosis will make it n. But mitosis preserves it, meaning keeps it the same. Keeps the same ploidy. And if you don't understand why, we'll see why in just a bit. So our main purpose for the cell cycle and mitosis is to allow the organism to grow, to repair and regenerate, and then also to maintain the organism. So for example, you lose a lot of skin cells, or you lose your red blood cells. Your red blood cells die off every once in a while, and so you have to replace those, and that allows you to maintain the organism and regenerate the organism. So those are the main purposes of the cell cycle and mitosis. You can see here a great picture of growth. There's a zygote, it becomes a child, that becomes an adult, and that's all through the process of the cell cycle and mitosis. Here we can see a limb being regenerated. So it was damaged, and over time through mitosis, the cells that were there were replicated and produced in the exact same way. So the cell cycle is essentially just a bunch of events that happens in a cell's life. It's the events that happen from it being a parent cell to it becoming a daughter cell. And the daughter cells can then become parent cells and then make more daughter cells, and those daughter cells can become parent cells and make more daughter cells. That just happens over and over and over again, unless your body tells it to stop, but we'll see that in more detail later on. So the cell cycle has two main parts. There's something called interphase. That's the growing phase. It's where you're preparing to divide, but you haven't divided yet. And then the division phase, where you actually divide. And that division phase is called mitosis and cytokinesis. So interphase is the growth phase, and the division phase is really mitosis and cytokinesis. Interphase has three parts to it, something called G1, S, and G2. That's where the parent is preparing to divide, so it doubles all its organelles, it doubles its DNA, so that when it divides, everything ends up being the same again, right? If I have $5, I multiply it by 2, that's $10, I divide it by 2, that's back to 5, right? So that's what interphase really is. It's growing, it's preparing to divide, it's doubling everything, so when it divides, you're not missing anything. The division phase, again, that's when the parent cell actually splits everything to both sides, then splits into two separate identical cells. It made a copy of itself, and now it needs to split into those copies, right? The copies can't stay attached to each other, otherwise it's useless. If I made a bunch of photocopies of something to give out to my class, and I stapled all 30 photocopies together, well then only I could have the copies, or only one student could have the copies, so I actually have to separate the copies from each other. So cell division, it makes it sound like it's splitting in half, but in reality it's actually making a copy of itself. So it is splitting, but through making a copy of itself. So the idea is that your cell starts off, let's say, with 46 DNA molecules, 46 parts to it. The cell during growth will multiply that by 2, so it has 98 DNA molecules, then it will split and make your 46 in each. So it starts off as 2N and ends up as 2N as well. If this doubling didn't happen, once the cell split, it would have half the amount of DNA. We'd have 23 and 23, and that's not what we want. We'd be missing DNA. When we repair something, we don't want missing parts. We want all the parts to be as they were. So here's a picture of the cell cycle. You can see this blue part over here represents interphase, which is the majority of the cell's life. It's actually 90% of the cell's life. And then this part here, the mitotic or division phase, that's just 10% of the cell's life. That's where the cell actually divides. It sends all the DNA to two sides, and then cytokinesis is when the cell splits into two separate cells. So in grade 10, what you basically studied is all this in detail. You saw mitosis actually happens in a few phases: prophase, metaphase, anaphase, telophase, and then there was a separate cytokinesis happening as well. So this interphase is growth, mitotic phase is dividing stuff. Mitosis is specifically dividing the DNA. So basically just sending this DNA to that side, this DNA to that side, like you see here. And then the cytokinesis is actually splitting the cytoplasm, splitting the physical cell into two separate daughter cells once everything on the inside has been sent to both sides. So we're going to start off by taking a look at the growth phase, and then afterwards we'll go take a look at the division phase. Again, growth, the main purpose is to prepare for division. It's to double things like organelles and DNA. And there's three parts to the growth phase or interphase. There's G1, there's S, and there's G2, and we'll study those in details. And then we're going to see the mitotic phase, which is the division phase, which involves mitosis and all its phases, and cytokinesis that kind of overlaps. So before we go into all the details, you can see the cell cycle. This diagram is shown up here in a cell form. Initially the cell has one DNA, let's just say one DNA, and it goes through interphase, and that makes 2 DNA. So it copied the DNA, it copied things. In mitosis, that's division of the nucleus. In other words, division of the contents of the nucleus, the DNA. So these 2 DNA are sent to opposite sides of the cell. So you get equal amounts of DNA going to opposite sides of this big cell here. But now we actually have to split the cell into 2, and so that's the cytokinesis part that results in 2 cells that have 1 DNA each. If we didn't copy the DNA, then they'd have half DNA each. We'd be missing some genes, and that would not be good for the organism that's trying to develop. So we have interphase, and then these 2 parts here, that's the mitotic or division phase, which is mitosis and cytokinesis. Mitosis, split the DNA, separate it. Cytokinesis, actually separate the cells or division of the cytoplasm. So let's start off with some details on interphase. Interphase, again, is the longest phase. There's no division happening here, it's just preparation for division, and it's 90% of the cell cycle. We're going to take a look at how you know you're in interphase. So a few things you'll notice is the DNA is in the form of chromatin. You'll notice there's a nuclear membrane, and you'll notice there's a nucleolus within the nuclear membrane. The nuclear membrane is just the nucleus, it's the membrane of the nucleus. And let's go over the 3 parts of interphase now. So in G1, the cell just duplicates organelles and cytoplasm, because each daughter cell should have the proper amount of organelles and the proper amount of cytoplasm. S phase is the longest phase of interphase. That's where the DNA actually doubles. So it's during S phase where the DNA is doubled. Now it's a bit hard to see here, but in reality your DNA is becoming doubled. So let's say you had 46 DNA. After S phase, you're going to have 92 DNA. You have 92 DNA. In G2, the cell will duplicate some more organelles. It will grow a little bit more. It'll duplicate these things here called the centrioles. In reality, the structure that looks like this is called a centrosome. And on the inside, these little cylinders, those are the centrioles. So in animal cells, the centrosomes contain centrioles. In plant cells, there's no centrioles, but they still have centrosomes though. So you'll notice that this one here is in G2. How do you know? Well, you can see there's 2 centrosomes. So I'm going to call them centrioles, but there's 2 centrosomes. They're still attached to each other though. Eventually they will split and move towards opposite sides of the cell. So DNA is in the form of chromatin, which is just uncondensed. It's not condensed, it's uncondensed DNA right now. It's been duplicated in S phase. Now, there's also some checkpoints that take place. So usually before going into division, the cell machinery checks to make sure that everything was replicated properly, and if there were any mistakes, then maybe we can fix them. If we can't, then there's other things that have to happen, and we'll take a look at what those things are. But basically, no division, just growth. There's 3 parts to it, G1, S, and G2. G1 is duplicating organelles and cytoplasm. S is called synthesis, and that involves duplicating the DNA. G2, that's where you duplicate other organelles and cytoplasm and grow some more, and there's duplication or replication of the centrioles, and then some checkpoints along the way to make sure that everything is being replicated properly and there's no mistakes in our DNA copying. And then some clues that you're in, let's say, interphase. You still have chromatin, you still have your nucleus, you still have your nucleolus. You might see these centrosomes are still attached to each other, but there's 2 of them. So remember I said there were checkpoints along the way. And so the checkpoints just make sure that everything is being copied properly, we have enough materials to continue with division, and if there is a mistake, we can try to fix it. Now sometimes you can't fix the error, so what happens is the cell goes through this process called apoptosis. And apoptosis has the word pop in it, is basically something called programmed cell death. That means that the cell kills itself. It triggers this whole cascade of events that leads to the cell becoming a blob and then breaking up in little pieces and then being broken up some more, and then it just stops reproducing. And I know it seems a bit dramatic, but if one cell has a mistake in it and it's allowed to continue reproducing, well then a whole bunch of cells will have a mistake in it, and that could lead to things like, let's say, cancer or developmental disorders. So if the cell can't be fixed and there's no other way, you have to do apoptosis for the greater good of the organism. And that's programmed cell death. The cell basically tells itself, okay, time to self-destruct for the good of the organism. So, a little bit more about that DNA replication thing I talked about in interphase. It's hard to see DNA replication, especially when you have chromatin. So I want to pretend like our chromosomes are more condensed. So let's pretend we're at interphase, and we have a cell that's 2n is equal to 4. What that means is you have 4 chromosomes, kind of like you see in the picture there. 4 chromosomes. They're not attached to each other. They're 4 non-replicated or non-duplicated chromosomes. Now in reality, I wouldn't see this just yet because they're still uncondensed in interphase. But after S phase, each of those chromosomes is copied. So this one will have 2 parts to it now, identical to the first because it's copied. This one will have 2 parts to it now. This will have 2 parts to it now. And so will this one. So in reality, after S phase, you have now 4 of these chromosomes. You don't really see them that way. You see them as chromatin still, but these are called replicated chromosomes. So this counts as 1 chromosome, 1 chromosome, 1 chromosome, 1 chromosome, but each of these is worth 1 DNA, 1 DNA, 1 DNA, 1 DNA. So let's call this chromosome, let's call this DNA. And these are also called non-replicated chromosomes. Now these ones here, after S phase, they're called replicated chromosomes. This is still 1 chromosome, still 1 chromosome, still 1 chromosome, still 1 chromosome, but it's 1 replicated chromosome. So we have 4 chromosomes total, but they're replicated. So what that means is they're worth more in terms of DNA. This is 2 DNA, 2 DNA, 2 DNA, 2 DNA. So if our original cell was 2n is equal to 4, after S phase, it now has 8. It now has 8 DNA. But still 4 chromosomes. The chromosome number didn't change, it's just duplicated chromosomes. That's all it is. So we're going to continue with that cell that's 2n is equal to 4 and track that movement. Okay, again, I was just kind of showing you, if you could see them as chromosomes at this stage, that's what it looks like. Okay. So here you can see that emphasized. So this 1 non-replicated chromosome is worth 1 DNA molecule. After replication, this replicated chromosome is still 1 chromosome is worth 2 DNA molecules. This is 1 DNA molecule here, 1 over here. They're identical to each other. Okay. Once they split apart, now you're back down to 1 chromosome, 1 chromosome, 1 DNA, 1 DNA. That's it. Just like the original parent was. So a replicated chromosome is 2 DNA molecules. A non-replicated is 1 DNA molecule. So just make sure you understand how to count these chromosomes. Here we have unduplicated or non-replicated, same thing. Here we have duplicated. This is 1 chromosome. This is 1 chromosome. This, though, is 1 DNA. This has been duplicated, so it's 2 DNA. 2 DNA. And these 2 parts, these 2 DNA, they're identical to each other because they're just copies of each other. We're going to label the different parts of different chromosomes soon. I just want to point out a few other things. these bands here, these little bands here, these are genes on chromosomes. So this might be a gene for eye color, this might be a gene for eye color, this might be a gene for hair color over here, this might be a gene for height rather, this might be a gene for hair color. We don't really know right now, doesn't really matter here, but scientists actually do know locations of various genes on different chromosomes. They've actually mapped it out. So this is what interphase would look like under a microscope. Under a microscope, you'll see a nice contained nucleus because the nuclear membrane is still there. You'll also see that the blue, which represents the chromatin on the inside of the nucleus, is nice and smooth. And it's nice and smooth because it's still in the chromatin form. When it becomes chromosome, it looks thicker, almost like worms, but here it's nice and smooth. It's in the chromatin form. I say it kind of looks like spaghetti, but you can still see the nucleus. And the centrioles, or centrosomes, they're the yellow. They're still kind of concentrated together. They haven't really moved apart yet, kind of like you can see here. So I want you to start picturing this microscope image like this cartoon here. Can you see the different parts in the microscope image? If you can start to see that, that means you're really starting to understand the process. Again, here's our summary of interphase. It's 90% of the cell cycle. Now what we're going to do is we're going to go take a look at the mitotic phase, so mitosis and cytokinesis. Remember that it's 90% of the cell cycle. And 10% is the division. This is just a little analogy to explain why the cell needs to duplicate its DNA. I did explain it in class. If you weren't there, just make sure you ask a peer about the analogy or come up with your own analogy to explain why DNA has to duplicate. Why not just chop it in half? A cell has 5 molecules of DNA. How many molecules of DNA will it have after interphase, and why? So here's our 5 molecules of DNA. After interphase, it should have 10, right? Because of S phase, that doubled the DNA. After the division, we should come back to 5. So the 2 daughter cells have the same amount of DNA that the original cell had. If we didn't double the DNA, then we'd have 2.5 DNA each. We'd be missing DNA, which is not good for development. Like I said, after division, it's time to enter the division phase, which is mitosis and cytokinesis, and we're going to take a look at all the different events that occur during this mitotic phase. Here we can see a summary of interphase. So this was interphase if the DNA was in the form of chromosomes, not chromatin. This shows our S phase over here. Now I want you to get that interphase is not part of mitosis. It's a different part of the cell cycle. Now we go to mitosis, where the DNA, the chromosomes, can be separated to both sides of the cell. And then we get to division part 2, which is actually physically splitting the cell into 2 cells. And so this mitosis part includes PMAT, prophase, metaphase, anaphase, and telophase. These are the main phases of mitosis. And so we're going to take a look at all of these in some depth. So we're going to start off with prophase. I have separated prophase into two segments, early prophase and late prophase. In reality, these steps, prophase, metaphase, anaphase, telophase, they don't just happen in one big jump. They happen in a more smooth fashion. It's not just prophase, boom, metaphase, boom. It's the chromosomes actually move from prophase to metaphase to anaphase to telophase, so on and so forth. So it's not a jump, 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 jump. There's early parts of prophase and later parts of prophase and early parts of metaphase and late parts of metaphase, so on and so forth. So early prophase, I want you to notice, that's this picture over here, the chromatin starts to condense into chromosomes. And the centrosomes, or centrioles as you see in this picture, start to move towards opposite sides of the cell. They go towards the poles. The poles just means the opposite sides of the cells. These little strings called spindle fibers start coming out of these centrosomes, because eventually those will attach to the chromosomes to start moving them, but not quite yet. You'll notice the nucleolus is no longer present. It's gone. It disintegrated. And in early prophase, the nucleus is still there. Still there. You'll notice this is early prophase because our centrosomes are still kind of close together. We still have our nucleus here, but the reason I know this is early prophase and not interphase is because you'll notice that the center is the DNA is no longer smooth. You can see these little worm structures. Those are thicker chromosomes. They're more condensed now. So now we'll go to late prophase, the picture on the right side. You'll notice that the chromosomes are now fully condensed, so they're condensed replicated chromosomes. The nuclear membrane is gone. It's been disintegrated. The centrioles or centrosomes have moved to opposite sides of the poles. They've actually reached the poles. They're at opposite ends. And the spindle fibers have attached to the chromosomes at the center here called the centromere. So the whole point is these spindle fibers need to attach to the centrosomes, to the centromeres of the chromosomes. And so to do that, the nucleus has to break down. If the nucleus doesn't break down, then you can't attach to the chromosomes, you can't move them. If you take a look at this picture under the microscope, you know that you're in later prophase because, look, these centrosomes have moved to opposite sides of the cell, and the DNA is no longer contained in the smooth sphere. And that little circle, all that DNA is broken out essentially. Now, students often ask, well, where are the other organelles during this? And they're still in the cell. We just don't really care about them when we're studying the cell cycle and mitosis. So there's still mitochondria there and everything else, but we don't really care about them too much. And also, what's the point of, well, condensing these chromosomes? When you think about it, it makes it easier to move. If you kept it as chromatin, well, you have a bunch of cords, a bunch of strings all tangled up. It's difficult to know what goes where. But when you condense it, it's nice and easy to move each chromosome. It's kind of like if you were moving from a home. Rather than keep all your stuff where it is, you would take it all and condense it into a box, and then it's more manageable. You can carry each condensed box to a different location. So I just want to point out here that we're still with our cell that's 2n is equal to 4, except we have our duplicated chromosomes. So our original cell in reality only had 4 DNA, but this cell here right now, because of all that duplication that happened in interphase, has 8 DNA. But there's still 4 chromosomes, right? There's still 4 replicated chromosomes. It's just that each one counts as 2 DNA. So now what's going to happen is those spindle fibers that have attached to the centromeres are going to start moving the chromosomes into a location that you might remember as being the middle of the cell. And we'll see that in just a few minutes. First, we're going to go and take a look at this idea of the centromere in a bit more detail. So here I've explained the idea of DNA counting. 2n is equal to 4. We have 4 replicated chromosomes, but 8 DNA molecules total. And each replicated chromosome is made up of essentially 1 DNA molecule that we call sister chromatids. So when the replicated chromosome has 2 of those chromatids attached together, they're called sister chromatids. They're attached together. We call them sister chromatids. They're attached together. The spindle fibers attach to a region called the centromere. That's where the sister chromatids are most closely attached to each other. It looks like the center doesn't always have to be right in the middle, but that's where they are most closely attached to each other, and that's where the spindle fibers will attach to the chromosomes. And essentially, you'll have 1 spindle fiber attached to each side of the centromere. So for example, 1 over here, and then 1 over here. 1 over here, and then 1 should make its way to here, and then 1 should make its way to here. Each one will have essentially a spindle fiber on both sides. So this is our replicated chromosome. You can see here is our centromere. Over here you have one of the chromatids, and over here you have the other chromatid. They're called sister chromatids because they're attached to each other, and they're identical to each other because they're basically copies of each other. They were made during the DNA replication during the S phase. Okay? this counts as one chromosome but is worth 2 DNA molecules. And you can see these ones under the microscope because they're thicker. As opposed to the chromatin, it's just a nice smooth smear. So during metaphase, what happens is the spindle fibers, those strings, move our chromosomes to the middle of the cell, a region called the equator or the metaphase plate. It's just an imaginary line that signifies the middle of the cell. So the metaphase plate, or also the equator or the equatorial plate, represented as the dotted line. There's no dotted line in the cell, but that's roughly what it's represented as. What I want to point out is that the spindle fibers that are attached to the chromosomes, they have a special name. They're called kinetochore microtubules. Kinetochore is essentially the region in the centromere where spindle fibers attach, and microtubules is just what the spindle fibers are made up of. So microtubules attach it to kinetochore. You'll notice there's some spindle fibers that are not attached to chromosomes. Those are called non-kinetochore microtubules. And they play a role as well. We'll see what that role is in just a bit. So by this point, the arrangement of all the microtubules and the centrosomes at metaphase, we call that the mitotic spindle. So this is this whole arrangement here is called the mitotic spindle. It's now nice and complete. Now, we're almost done. We've set up the chromosomes right in the middle. The goal of doing this is so that we can now split the chromosomes apart and get equal amounts of DNA on both sides. If you take a look at this picture here, you can see a chromosome with some spindle fibers attached at the centromere here, and that region in the centromere where spindle fibers attached is called the kinetochore. So this is a kinetochore microtubule. Here is a picture under the microscope. You can see the centrosomes over here. Don't confuse those terms, centrosome and centromere, very easy to confuse. Make sure you go over them. And here you can see the thick band of chromosomes right concentrated in the middle there, which means that we're in metaphase. So again, if you had to count some DNA here and some chromosomes, you still have 4 chromosomes, 1, 2, 3, 4 replicated chromosomes, but it's still 8 DNA because each replicated chromosome is worth 2 DNA molecules. Now we get to anaphase, where the replicated chromosome is split apart. So the sister chromatids are separated from each other. The way that happens is that the spindle fibers, the kinetochore microtubules, they shorten. Kind of like if you ever went fishing, or if you ever, let's say, want to attach a rope to something and you want to pull it in, you pull on that rope and you shorten the rope, the distance between you and the object. So the spindle fibers, the kinetochore ones, they shorten, and then that causes the sister chromatids to separate. And so now your chromosomes are non-replicated again. They become non-replicated chromosomes. So technically in this cell, we have 1, 2, 3, 4, 5, 6, 7, 8 chromosomes, but still 8 DNA molecules, right? So our chromosome number has changed only because of the definition of the chromosome. It's no longer replicated, they're non-replicated chromosomes now. But the idea is that 4 go this way and 4 go this way. The cell has been elongated as well. It's more of an oval shape. It's not just your imagination. And that's caused by the non-kinetochore microtubules kind of pushing off of each other. And that just makes it a little bit easier to separate the chromosomes. So that's why the cell is elongated. Here you can see a picture of the chromosomes during anaphase. You can see the band is no longer concentrated in the middle, but rather that band has been separated. And so the chromosomes are moving towards opposite sides of the cell. And that's during anaphase. We've already done chromosome counting, but again, now we have non-replicated chromosomes. We no longer have sister chromatids either. They were only called sister chromatids when they were attached to each other, as in this case here. Now that they're separated, they're technically no longer sister chromatids because they're separated. They had a fight. They don't call themselves sisters anymore. There's still 8 DNA molecules only, though. It's just that now they're on separate chromosomes. And this is just, again, to emphasize that idea of replicated chromosome with sister chromatids, split up during anaphase. They're no longer sister chromatids. It's still worth 2 DNA. It's still equivalent in terms of what the DNA looks like. This DNA here and here is the same as the DNA on here. So now we get into our last stage, which is telophase and cytokinesis. Telophase is technically the last phase of mitosis. Mitosis was just about moving that DNA to opposite sides of the cell. So in telophase, essentially our DNA has reached opposite sides of the cell. A nucleus is going to start to reform. You can see the nuclear membrane reforming around the DNA to protect it. The nucleolus starts to reform too. And our chromosomes are becoming chromatin again. They're becoming chromatin, but the idea is that you should have 4 in here, 4 in here. And while telophase is happening, this overlapping phase called cytokinesis happens. That's division of the cytoplasm. It's actually splitting the cell up into 2 cells. So there's this little ring of muscle fiber that pinches the cell into 2. And so the reason I can tell anaphase and telophase apart from each other is because usually in telophase there's a few hints. First of all, you can see the cell pinching inwards, and then you can see that the DNA is becoming smooth again, like chromatin. So I can see those hints that tell me that this is telophase as opposed to anaphase. Because in anaphase, I wouldn't see that pinching inwards, and I wouldn't see the smooth DNA, whereas in telophase I would. I'm going to talk about cytokinesis in a bit more detail in the next few slides. But before doing so, I just want to emphasize again that these daughter cells are identical to each other and to their parent. They're the same of everything from the parent. They're copies. There's no difference. So what mitosis did is it preserved DNA, it preserved ploidy. It didn't change that. It kept it the same. Now these 2 daughter cells can become parent cells and then go through their own cell cycle if that's what the body wants them to do. If not, they might get a signal to do something else. They might get a signal to specialize. They might get a signal to maybe destroy themselves because there was some mistake somewhere along the line. There's so many different things that can happen with these cells. So in cytokinesis, just to go over it in a bit more detail, in animal cells, you can see this pinching that happens. This little indentation that forms is called a cleavage furrow, and it's formed by this group of fibers called a contractile ring that just pinches the cell into 2 eventually. Whereas in a plant cell, there's no contractile ring, there's no cleavage furrow. Instead what happens is there's this thing called a cell plate that starts to form between the 2 cells, a cell plate. And the cell plate usually forms from little vesicles that come from the Golgi body, one of the organelles in the plant cell. And typically after that's over, the plant cells that were produced kind of stick close together. That's generally how plants work. So there's no cleavage furrow in the plant cell cytokinesis. It's just really a cell plate forming, and then they kind of stick together. Whereas in the animal cell, there's a cleavage furrow with a contractile ring to pinch the 2 cells apart from each other. That's just one of the main differences between plant and animals mitosis. There's also the fact that plants don't have centrioles in their centrosomes, but they still kind of operate the same way with the spindle fibers. Okay, and here you can see a microscopic picture of animal versus plant. You can see that little cleavage furrow form here, and over here you can see the cell plate forming in between the 2 plant cells. So this is the other difference. Again, plants don't have those centrioles, but animals do have the centrioles within them. So you should know those main differences between plants and animal cells, cell cycle, mitosis, cytokinesis. Hopefully you understand this joke now. And this is just a little summary of all the steps together. I recommend that when you study the cell cycle and mitosis, that you study them first as a whole. Take a look at how all of them go together. To study them in terms of their cartoon image, but also in terms of their microscope image, to see if you can make the parallels, to see if you can see what the different parts are. I would practice drawing them out. So practice drawing these stages out with maybe 4 chromosomes, maybe try 6 chromosomes if you need to, or 5 chromosomes, and see if you can end up with the same amount of chromosomes in the end. You only really know if you understand it if you can draw the entire cycle out. Again, here we can see the latter parts of it. And here we have our summary of our identical daughter cells. identical to the parent cell that we start off with. So the next few slides are just an animation of the cell cycle. There's some great videos out there. I highly recommend that you go watch the videos to see how it all fits together. I always like to maybe study a process without a video first so I can leave it up to my imagination, and that way it makes my brain work a bit more and I can really commit that to memory. But afterwards it's okay to watch a video or an animation. So you can take a look at this animation. So we saw our chromatin duplicate. We saw more stringy stuff was produced. Our centrosomes were replicated. Now we're going to prophase, where chromatin condenses to form chromosome. Our nucleus disappears, and our centrosomes move to the poles. Spindle fibers attach to the centromere of the chromosome. The chromosome is moved towards the center of the cell. The spindle fibers shorten and separate the sister chromatids from each other. And then telophase and cytokinesis will occur to produce two daughter cells identical to each other and to the parent. And don't forget that these will become chromatin again. This is a similar animation just along the cell cycle diagram that you saw. So our cell goes through interphase, it grows, doubles its DNA. Now it goes into the mitotic phase, prophase, metaphase, where it lines up in the middle, anaphase, where the sister chromatids are separated, and then telophase and the overlapping cytokinesis to get our two daughter cells. Now there is a bit of an error in this animation. I didn't get to fix it. Hopefully you spotted it. Maybe you can pause the video and figure out what the error was. It's a very small detail, but it is still an error. Hopefully you noticed here there shouldn't be two centrosomes yet. They only replicate really after that G2 part, not G1. So I should have had one and then have them replicate in G2. There's a great video here that you should watch. All you have to do is click on the link through the original PowerPoint and you can see what the video is. It's a great way to summarize that knowledge and understanding of the processes of the cell cycle and mitosis. And again, it's a great way you should always study different pictures to really get a good idea of the steps. You don't just have to memorize the same picture that you have in the PowerPoint. You should be able to identify the different steps from different pictures or from different from different images of microscopes, right? So you can see, for example, maybe some interphase here. You can see prophase with thicker chromosomes. You can see a metaphase over here because the band is lined up in the middle, and you can see the centrosomes over here. So eventually all this will start to make more sense to you and will become much clearer to you as you start to understand all the different parts of the cell cycle. And it's quite amazing when you start to see these images and can make sense of them when you couldn't make sense of them before. Again, just to show you, here I have some anaphase and here I have some telophase. They're sometimes hard to tell apart. Usually the anaphase, they have more of these little finger-like projections pointing in the opposite direction of where they're going to, because the chromosomes haven't been fully condensed yet. And then here, fully decondensed yet into chromatin, sorry. And then here you can see that we're more in the chromatin form. And you can also see that cleavage furrow over here too. So those are little things that you can kind of start to look for as you try to determine what phase of mitosis or the cell cycle that you're in based on the microscope images. And it's okay to sometimes say, for example, that this might be late anaphase, maybe early telophase, but still I would say the best answer here would be anaphase because I don't really see a cleavage furrow forming yet. So very quickly we're going to take a look at what would happen if mitosis went wrong. So if mitosis does go wrong, let's say it stops prematurely, it stops before it should, then you might have some developmental disorders. You might not grow properly, so you might have maybe partial organs or partial limbs, or you might not be able to heal yourself well when you're injured, and many drugs actually cause that. So in history you might learn about the drug called thalidomide or Agent Orange, which actually affect the cell cycle of developing organisms and stops mitosis earlier than it should stop. And so you'll notice that you have these limbs that haven't been formed properly or haven't been formed at all. So this cell cycle stuff that we're reading, that we're learning about, has some really, really serious implications and applications for your life, literally. So it's really important to understand this at a biological level. When scientists create medication to treat people with diseases and disabilities and to prevent diseases and disabilities, they need to understand what's happening at the cellular level so that they can treat the cell so that it can grow properly and it can divide properly. These are some shocking images, but sometimes mitosis needs to stop. Eventually you grow enough and your cells need to stop dividing. And they usually get a signal that says, hey, stop dividing, that's enough. But sometimes your cells don't listen to that signal and they just keep dividing and dividing and dividing and dividing, even though we told them to stop. And that usually happens because there might be a change in the DNA that happened by accident. It could have happened maybe just randomly, or maybe something caused it, like some dangerous toxins from cigarette smoke or from pollution. And it could have caused a change in your DNA called a mutation. And that mutation basically makes your cell ignore your body, ignore signals, and just keeps dividing and dividing and dividing. And when that happens, eventually, your cells will form this huge mass of cells called a tumor. And if that tumor essentially travels from one part of the body to another, it can become cancerous. It can become a cancer. Sometimes the tumors are okay, they're safe, meaning they might just cause some discomfort, but they don't move and they don't go anywhere. But if the tumors do move, then they can be cancerous, and that's a whole different story from the non-cancerous ones. But the idea is that if these mutations happen, your cells stop listening, cancer can happen. And cigarettes and certain other toxins can lead to these mutations. So you just need to be careful, and you need to have a good understanding of what can lead to mutations to maybe prevent them as well. Mitosis has some really cool applications that I do want you to read about, particularly cloning. Here we can take a look at a very simple cloning process that that we discovered in 1958. Essentially, we took some cells from a plant, from a carrot, put them in these little petri dishes. We gave them enough nutrients, they started to grow, and they made clones. They made clones, they made more carrots. So this is one way to clone a plant. Very straightforward application of mitosis. These two carrots would be genetically identical to each other. You can also clone animals. This is something that we're still working on, but this is the first clone that was created. You might have heard of the sheep named Dolly. So essentially, here's what happened. You had a donor cell. We took the nucleus out of that donor cell. We had an egg cell donor. So let's call this the mother. We took the nucleus out of this egg cell. So we had an empty egg cell. We put the nucleus from this sheep over here into the empty egg cell. And what happened was we applied an electric shock to it, and this cell started to divide and divide and divide and divide, and eventually it became an embryo. And we then put this embryo into a foster mother, and that embryo eventually became the cloned lamb. So just once again to highlight what happened, we took an egg cell from a donor, took out the nucleus, took a donor nucleus, put that into the empty egg cell, applied electric shock to it, it divided and divided and divided to make an embryo. We put that embryo into a donor mother so that it could grow and develop in there. And then when it gave birth, it was the cloned lamb from this sheep here. They were genetically identical to each other. And that sheep became known as Dolly. So that was an application of mitosis. And I do want you to read up on this a bit more in your textbook. So the pages that you should read up on for this topic are 143 to 151. The questions you should do are questions 1 to 5 on page 151. And there's other worksheets you'll be provided with, and also a few class activities that you should do as well. You should also watch some videos and practice drawing mitosis, as it will really help you to understand the process of meiosis.